These are the results to our genetic recombination lab that we just completed in AP Bio. We used E. coli bacteria. We inserted a bacterial plasmid called p-glow, which did a couple of things. It made our E. coli antibiotic resistant to ampicillin. It made our recombinant bacteria, the bacteria that took up the plasmid, fluoresce under ultraviolet radiation. So we're going to take a look at that. So if we got successful results, we would have bacteria that light up when you hit them with UV light, and they're not killed by an antibiotic. This process is called genetic recombination. Recombination means that we're putting in genes from an unrelated organism into an existing organism. That's what recombination is. So that's kind of the goal of this lab. So as we're watching the video here of our results, we're going to have 3 plates. The plate all the way to the left on the video is it's a nutrient agar is all that's in the plate, basically just bacteria food and nothing else. So all bacteria should grow on that. Ones that have been genetically altered and normal standard E. coli will grow on there equally well. So everybody grows on that plate. The middle plate has is nutrient agar that has an antibiotic mixed in with it. Ampicillin is mixed in with the nutrient agar. So in the middle plate, only our genetically recombined organisms should be able to grow, because they're the only ones that will have the gene that allow them to be antibiotic resistant. So what that means is any bacteria that didn't take up the DNA that we introduced is gonna die, and only the individuals that took up the plasmid that we introduced survived. And then they're going to reproduce over and over again and make millions of copies of themselves, including our engineered gene that we gave them, and all of their offspring will exhibit the same traits. In other words, all of their offspring will be able to live on the antibiotic plate. And in the last one, we had the nutrient agar, we had the antibiotic, and in addition to that, we had arabinose sugar, which is in this particular bacteria that we made, that arabinose sugar allows the glowing enzyme to be expressed. So it's an epigenetic interplay there, that the presence of arabinose sugar turns on a gene that allows the expression of the fluorescent protein in the bacteria. So in the third plate, all the way to the right, we would expect to get these glowing bacteria under UV light. So let's watch the video real quick, and you can see what happens here. Those are all 3 of our plates. And I tried to close up here. The camera I'm using is just a little flip camera, and the resolution's pretty poor, so I'm going to have to explain what's going on here. But what we see on that first plate is just a mat of bacteria. The second plate, you see a lot of air bubbles in the gel because we had a rough time pouring the gel, but there's a few little spots there of actual growing bacteria. And finally, in the last plate, you also see some spots of growing bacteria, but it's not a mat, because most of them have been killed because there's antibiotic in those 2 plates. Now when the lights go off and I hit them with UV radiation, the first plate, nothing significant happens. The second plate, nothing significant happens. But in that third plate, when I get in close here, you can see the fluorescing colonies of bacteria. They actually light up. And so that was kind of the point of the lab. We were successful. We introduced genes to our standard E. coli that make them produce a fluorescing bacteria, or a fluorescing protein within the bacteria. So we were successful. I was worried that this lab wouldn't work out, but it did. It worked out quite nicely, and I'm very happy with it.